I need to press this button and wait for it to start.
We're going to do it's going to be pretty quick and informal.
Okay, we're ready to go.
Give me a second. I'll just record ours.
All right, ready?
Yeah.
So what we're going to do right now is, since we have a bunch of new people here, we're going to do a very quick, Super and I thought about doing a set of little mini tutorials about neuroscience. And I think we both thought that a good place to start would be to talk about neurons and how they work. So they can be very interactive. I kind of know this stuff, but I haven't prepared anything. So so just you can help direct it as we go, okay? And it's really, you can sort of paint two worlds here. You have what are classically called the point neuron. At least it's called that way in some worlds, which is the sort of classic neural network neuron from a long time ago. And that looks like this. You have a bunch of inputs. Well, these are synapses. Each of these has some sort of weight. They all get summed together somehow. That's supposed to be a signal. And then there's typically some sort of nonlinear output function on this. And so you have an output of this thing. And so each of these has some sort of scalar value that comes in. It gets multiplied times the weight. You sum them together. You put it through some nonlinear function, and then you get another scalar output. That's your classic point neuron model. And then we'll contrast that to, in biology, in the brain, there are many different types of neurons. They're not all the same. 80% of the neurons in the brain are excitatory neurons, meaning those are where most of the sort of, one way to think of it, that's where most of the processing occurs. And then 80% of those are pyramidal cells, and the other 20% are called stellate cells, but they're sort of viewed as a subset of pyramidal cells. So I'm going to talk about the 80% of the cells that are excitatory cells, and then we'll talk about the inhibitory cells in a moment. So the pyramidal cells, we typically draw them with a little pyramid shape, because the cell body looks like that. That's why they're called pyramidal cells. And we'll talk about how they work.
Is this a subset of that 80%, or is this?
But this is 80% of the cells are excitatory. 80% of those are like these. Okay. But the ones that aren't like these, the 20% that aren't like these, are kind of like these minus that. So it's like they're. This is called the. I'll explain that in a moment. But let's just put it this way. So this is a pyramidal neuron. And one way to think about it has been argued that the majority of the other neurons that aren't pyramidal cells are really just like pyramidals. Like this, the next biggest category is called the spiny stellate cells, but they're sort of like pyramidal cells, but they don't have this apical dendrite, and so they don't have that little pointy hat thing as much. And so it may be there's evidence that this is just a derivative of this, and they work actually on the same way. Okay, so I'm just going to talk about pyramidal cells for the moment. Now, there's a lot of things we can talk about here. The differences between these two. First, let's just talk about the synapses for a moment. Okay, pyramidal cells have lots of synapses, thousands and thousands of synapses. Some have up to 30,000 synapses. Most they don't really know, but it's estimated somewhere between 3 and 10,000, something like that. It depends where they are. They're all doing the same. The synapses are none of them are on the cell body, none of them, and they're arranged along the dendrites. And these are called the basal dendrites, and this is called the apical dendrites, and this is called the apical shaft. And so all the excitatory synapses are along out here, and none of them are on the cell body. None here. There are inhibitory. Now, one thing about these synapses, here a synapse can have a plus and a minus value, like they can be positive or negative. Here, these are only pluses. You don't get any negative synapses. There are a second set of synapses which are negative synapses, inhibitory synapses. And I'll show those of green. And the inhibitory synapses are they're very, very different. They work differently, and they're at different places. So there's a set of them. There's three classes of them, and generally there's some that are lighter along here. This is called if you don't know this, I'm sorry. I should tell you. This is called the axon, and these are dendrites. And of course, the axon goes out and then branches and connects to other synapses. So this is the output of the cell, and these are the inputs of the cell. So the axon is equivalent to this, and these are equivalent to these guys. So there are inhibitory cells, inhibitory synapses right around the beginning of the axon. There's inhibitory cells that are on the cell body, and there's inhibitory cells that are spread along here.
Inhibitory cells or?
I mean, skin to synapses. I'm sorry, inhibitory synapses are spread along here. Most of the synapses in the cell are excitatory, so far more, right? But the inhibitory synapses in general are believed they look different, and so at a first order approximation, you could say that learning occurs with the excitatory synapses, and the inhibitory synapses are not learning anything. They're there for regulation and function. So, for example, the way an output of a cell happens in a neuron, you get this spike, right? It's a single spike, and spikes are spikes, so they all look the same. And so a spike will be a transitory voltage transient that travels down the I don't know how many, if you know this stuff at all. No?
I've read the papers.
Okay, so while you go into how this occurs, I mean, these are biological cells of all kinds. I can explain how this all occurs, but what happens is when the cell body gets enough input, it can generate a spike, and the spike then travels perfectly along this thing out to all the extremities here.
So I have a question. So the inhibitory synapses,
Yeah.
you said they're the only ones who exist in the cell's body,
Mm-hmm.
and do they connect to other cells' bodies, to the body?
They come from other neurons,
Yeah, but from dendrites or from their body as well?
axons.
Axon. Oh, okay.
The output of every cell is an axon. There's a few exceptions to this. So there are a few places where there are the dendritic axons, but right now for today, just say the output of every cell is an axon, whether it's an inhibitory cell or an excitatory cell. And then the inhibitory cells make inhibitory connections, and so there's another class. The way to think about it is the inhibitory cells and the inhibitory synapses are more like regulation or plumbing or code. They're not where most of the learning occurs. So learning occurs in the excitatory cells, and so but there's a complex function of how these things interact, which is largely based on the inhibitory cells, how the inhibitory cells connect and control things. So I was going to give you an example here. Again, what happens is when the voltage, this doesn't really matter from modeling point of view, but when the voltage of this cell, if you look at the internal voltage, here's 0 volts. The voltage is maintained down at a negative 60 millivolts. That's a sort of standard point. And then it raises when he gets inputs. It goes up a bit, and if it gets above a certain threshold, then you get this spike, and it goes back down again. Okay, so that's what's going on here. It's like summing these inputs in some way, and then it gets enough. It goes, oh, a spike, and the spike goes whoop, down here and travels along, and it goes to other cells. Well, these inhibitory synapses right here are ideally positioned to stop the cell's spike from going down the axon. It's like saying, yeah, you may get all that input, but I'm not going to let you send out a spike. So it's a control function. It's like a throttling function. It's a gate in some sense. It's not it's not learning anything here. It's just saying another cell.
Maybe for sake of
First order approximation. One of the rules about neuroscience is every rule there's an exception to every single friggin' rule. I mean, so Marcus knows a lot of this stuff, and so I'll say that, well, yeah, and then there's someone found the cell where this did this, and yeah, I know, but.
Even the synapses aren't learning.
Well, the inhibitory synapse is the first order approximation. Some do learn, but you could think of them. Most of them as not learning.
But the cells that they're coming from, the cells that are inputting into the synapses, they're learning.
Probably.
They have dendrites.
Yeah. Remember, there's like.
That's a good point.
It's a good point. you know, we could argue there are many dozens of types of neurons that have different learning rules and different functions and so on. So it's a very complex thing. There isn't like one type of neuron. You can take the neurons and divide them into excitatory and inhibitory. Then you can divide those into thing, you know. So there's many classes of inhibitory neurons. Actually, you could argue there's really two basic classes of excitatory neurons, and I have already said they're almost the same. You know, almost all the excitatory cells can be either pyramidal or spinal stellate, and those are really basically the same. But in the inhibitory world, there's a dozen different inhibitory neurons. They're very functionally different. They have different morphologies. They look different. They have different rules they work on. So there's much more excitement going on in the inhibitory world, which is only 20% of the neurons. But most people don't know how to model any of that stuff. They don't know what it's doing, so they just focus on sort of this kind of model, which is the excitatory neuron.
Yeah, I think like it is useful to know whether it's reasonable to use negative weights in our, like, when we're doing these machine learning algorithms. If we use negative weights somewhere, does that just throw biology out the window? Or can we think of that as like abstracting away the fact that these inhibitory neurons in fact do learn?
I think you could say that on an individual cell, having synapses that go positive and negative is out the window. Okay,
Yeah.
That can't happen on a biological cell. For the most part, these inhibitory synapses do not learn. For the most part, there are exceptions to that. So I think this to start off with, but you definitely don't get positive and negative. But that's not even the worst of the problem at the moment. We get to the worst of the problem at the moment. So we have this very different world where we have lots of excitatory synapses that can go from 0 weight to some value, and we have 20% or so of the synapses are inhibitory, which don't seem to learn at all. They're there for like controlling things, controlling thresholds, and it's sort of like the code of the network. How do you implement, how do you implement boosting, or how do you implement sparsity, right? So let's stop that. That's talking about the distribution of synapses on these guys. The next thing I talk about what kind of synapse learn. Okay, over here in this world, we assume these guys are usually some sort of floating point value, right? And we also assume that the output of this thing is a floating point value, some number of digits of precision. What's going on over here? Well, you could say, well, I only get this spike on the output. So you say, well, yeah, but the spike rate can change, right? So I can have a fast spike rate, I can have a slow spike rate, and therefore I'm encoding a scalar value or floating point value in that. There is some truth to this, that if you look at many of the neurons in the brain, they do exhibit faster spiking and slower spiking. There are many neurons in the brain that don't seem to do that. And in contrast, but this is true, but there's also some very conflicting evidence. So for example, if someone is given a task to say, okay, I'm going to show you a picture of a hippopotamus or a unicorn, and you have to push the button, the right button or the left button, whether you got a hippopotamus or a unicorn, right? Well, you can do that really fast. And you can do it fast enough so that they say, okay, here's the eyeball, and here's the axons coming out of the optic nerve coming out of the eyeball, and then it goes to this neuron, then it goes to this neuron, it goes to this neuron, it goes to this neuron, and you can count up how many steps it takes before you push the button, right? And if you take those steps, there isn't enough time to get the second spike. So on some tasks that are cognitively difficult tasks, you cannot be doing time averaging of the rate. It's just not possible. There's not enough time to do it.
The spike rate can be very high, right? We can have like 100 hertz.
It can be, but even 100 hertz, it's just not enough time.
But you can't respond faster than what, 200 milliseconds or something?
Yeah, but you have to add up all these steps. Right. This has been done. I mean, these are published papers, so there you go. It's like Well, typically 100 hertz is very fast, and so you have to look at the neurons in the visual cortex. And a typical fast response there might be 50 hertz. So you got 20 milliseconds, and then if you have 10 steps before you can push the button, then that's 200 milliseconds. And there you go, and you're doing it in 200 milliseconds. It's that kind of analysis.
So you're saying like rate coding biological.
It's not always necessary. I'm not saying it doesn't happen. It does happen. It appears to be important in many different places. When I exert a force using my muscle, there are spikes coming down to the muscle, and the rate of the spikes corresponds to how tense my muscle will be. And I have this thing I do at night sometimes. I don't know if anyone else do this, but I sleep on my head on the side. Often the pillow is pushing up against my ear, and I can hear. Hear me out on this one. I can hear what's the individual spikes going to a muscle in my jaw. I don't actually hear the spikes. I hear the muscle. But it's a very clear rate coding. I go, and I can go up and down by tensing my jaw over and over again.
I can hear it's noise, but I was assuming it's like.
No, but that's a different sound. That's a swoosh, swoosh sound, right?
Yeah.
This is I get this maybe once a month. I just happen to be just right. And you can it's really fun. You can just try to tense your muscles. And what I'm hearing is the spike arrives at the muscle. The muscle does a little miniature contraction. That's what I'm hearing, of course. I can't hear the spike. But anyway, rate coding, it does exist in the brain. But my point is that many cognitive tasks do occur quicker than you have time to do the rate coding. So I'm not saying there isn't rate coding, but I'm saying there's a lot of things in the brain which don't rely on rate coding. It's just you can do hard things that there really isn't any time to get any precision. It's when the first spike occurs is really what's important. And so there's this is a fact. And so it's not to say that there's no rate coding, but we also said, hey, I got to understand how a system can work where I can't rely on rate coding here in many situations. And you look at the models we did with our HCM temple memory model, it really is relying on the timing of the first spike is what it really is about. That's the key item. So here you have this scalar output. Here you can have a scalar output, but many cognitive functions occur without it. So you certainly have a lower resolution. You can't rely on any kind of high precision here. In many cases, it's just the timing of the first spike. So we have to keep that in mind. That's a big difference. And then also we look at the weights of the synapses. Now here in these point neuron models, there's an assumption that you have a fairly high precision in the synapse. Real synapses aren't like that at all. A real synapse looks like this. You have a dendrite. You can take one of these little synapses here. You have a dendrite, and the synapse, it kind of looks like this. It's typically on a little spine. And I'll draw two of them. And then here so this would be the dendrite, the black line here, and I'm just going to draw in green an axon that's making a connection to this. Okay, so I might have an axon coming along, and as that axon passes by, it makes a connection onto here. And another axon comes along, and it makes a connection onto there like that. Okay. So this would be a spike would come down here, hits this thing, and then what happens is it transfers to this guy, right? So that's like an input coming in, and then it hits one of these black synapses, and then it's what's the effect on the other side? Ever follow that? So just a couple things here. What happens is technically, and you don't really care about the technically what's going on. You have this spike that comes in. I'll just run black. You have the spike that's traveling down here. When it gets to here, there's these little vesicles. These vesicles spill out some chemicals into this little gap, and then they migrate, they diffuse across the gap into these little receptors here. And what basically occurs is that when this spike arrives here, based on how many of these vesicles are released and picked up on the other side, the voltage lowers here. So you get the voltage goes down a little bit locally here, right? So we're trying to depolarize this cell over here. And the idea is if you depolarize this cell, then you can get enough of that input that this guy will spike.
So voltage goes up, it's depolarizing.
Yeah, the voltage, yes, it goes up, yes. Yes.
Polarizing.
torus is very.
Yeah. It's at a minus voltage and it goes up towards the threshold. That's not really important for the modeling point of view, of course. And now, what can we say about this? There's a couple of things you can say about it. First of all, the amount of chemical released here is very variable. It is low precision. In fact, what can happen is after this thing's released some, another spike can come in some synapses, there's nothing left to release. It does nothing. It's like I already depleted myself. Sometimes it'll get a bit more. You say, oh, I got some up here. Sometimes it's a very stochastic process. It depends on who's around, how many of these are vesicles, where are they, are they close to the surface, So over the years, it's become very clear that there's very little precision in this, almost none. Yeah, it's described as highly stochastic how much chemicals released. Now, it's not true that it's completely stochastic. Some of these synapses, I've drawn two here. This is a very robust one. It has a big surface area there. This is one that's called a philopedia, it's called. This would be like a newer synapse, and it has a smaller area here. And so it can also release chemicals across the gap, but it's probably going to release less. But this could also release nothing too. So I read a paper once, and I've never been able to find it. I read it many years ago, where someone was looking at this and says, you know what? We can't really assume that these synapses have much value to them. It's not like they're like you can have 2 or 3 digits of precision, or even 1 digit of precision. It doesn't seem possible. They said, but it looks more like the difference between a synapse which is old and heavily learned and one which is new and not so learned is more like permanent. It's like, so there's some synapses that are really hefty like this, and they kind of stick around for a long time. But there's lots of synapses that are kind of like little leaky things, and they can come and go all the time. In fact, it's not common, I mean, it is common that a new synapse can grow very rapidly. In a matter of hours, you can have a new synapse come out here and form out here. And so the idea is like, we can't really have high precision here, but we can form new ones rapidly, and they can go away rapidly. And it's more like you really can't rely on the precision of it in any sense. So we decided here at Numenta, we're going to model these as binary. And so it's either we're going to say the synapse is there or the synapse is not there. But if it is there, it's just going to be a 1. We're not even going to try to, you can't rely on 2 digits or 1 digit of precision. That's not really accurate. But what's more important is that these ones are forming or not forming, and some are more permanent than others. And so let's go with that. And it means also that any individual synapse really can't be very useful on its own because they're stochastic. So you can't rely on them. They're all kind of loosey-goosey. So it's always going to be a population of synapses, which is important. Yeah.
How would you measure the permanence of the brain? Is it like the size of the synapse?
Well, that's the simplest way of looking at it. These are very complex biochemical systems here. They have lots of things going on. There's all kinds of receptors and neurons, so proteins being released and so on. The simplest way to look at it, so there's different types of receptors in the surface here, but the simplest way to think about it as a synapse gets reinforced, like with heavy learning, it moves from being this very thin little thing to a really robust thing like this, and the physical structure here is harder to get. You can look at it and say, oh, that's a robust, bouton, they call it, so bouton.
Okay.
And so it's not going to go away tomorrow.
And when you grow a new synapse, how do they work? So you just grow to connect to.
No, well, so the basic theory here is the following: is that you have axons that, like in the green stuff here, and you have dendrites, the black stuff here, right? And they're all coursing together all over the place. It's a big pile of spaghetti in there. And any individual synapse, neuron. If I look at this neuron, I can say, well, it can only form synapses to axons that pass somewhat near its dendrites. If there was an axon that was passing out, out here, it can't make a synapse to that. But if there's one that's coursing through it like this, different places it can make synapses. It's called it's a potential synapse.
Is it like the closest one?
Yeah.
It just grows.
Well, it has to be close.
Okay.
but not the closest. So we think there's some ruling rules we'll talk about in a moment here. But in the brain, it has to be close. So we call this, it's not a new word, we didn't make this up. You can think of all the potential synapses this cell can make. In the brain, you can imagine in the brain, imagine I take a square millimeter by 1 millimeter by 1 millimeter, let's say by 100 microns, a tenth of a millimeter. In that little block, I might have 10,000 neurons, okay? And in here you might have, hundreds of meters of axons and dendrites. It's really like super small. These things can be as small as like a micron, thousands of a millimeter. And it's just like, if you see pictures of this and stuff, it's just all, they can reconstruct it, just totally all over the place. And so at any point in time, a neuron it's going to have a lot of other axons passing nearby, and it can make connections to them. But if the axon doesn't go close, it can't make a connection to them. Now, complicating that is the following: if there are active synapses here that are useful towards the end of this dendrite, the dendrites will continue to grow. They'll branch out as if they're going to try to find more connections. So in software, we don't have to do any of this. In software, we can say our neurons can potentially connect to any other neuron, but in the physical brain, they can't do that. In the physical brain, they have to be lucky to find the right things to connect to. And so they have to constantly try to find things that they might connect to in a heavy and learning way that are useful. But we don't have to model that, okay? Early on here in Numenta, we modeled these potential synapses, and we said, okay, there's only so many neurons. This neuron can only connect to these neurons. And then after a while, we said, we don't need to do that anymore. We'll just say, there's 10,000 neurons. If I want to train one of these neurons here, I'll just randomly pick from the other 10,000 which ones I want to connect to. We don't have to worry about this physical constraint of proximity. But the important thing here is that the synapses they're not scalar values. Everyone still thinks of that. Evidence is they're not.
So one thing I've heard people bring up, and I don't know how widespread this is, but so I don't know how widespread this is, but what if this axon connects to multiple spines on the dendrite in a small area?
Yeah. Do you remember this conversation we had about this? It doesn't happen.
Okay.
There's a few exceptions where it does happen, but rarely it happens. It certainly happens with inhibitory synapses. An inhibitory neuron can make multiple connections on the same cell. Because basically I'm saying, I'm going to shut you down. I have all this input on you. I'm going to clamp on your head. You're done. But what you find is if you look at one of these axons and it makes a connection here, it won't make 2 or 3 in a row. It just won't do that. And there was another paper I read, and I have not been able to find again. Someone made an analysis of this, and they found a very clever mechanism that occurs in axons, which prevents them from making multiple synapses near each other. And there's a reason for this. Is it obvious what the reason for this is? No. Imagine you have a population of active cells, a sparse population of active cells, and you have another sparse population of active cells. They could be co-located. They could be the same cells. They could be remote in the brain. And what you want to do is you want to have this guy invoke this guy. All right? I want to have this pattern A, invoke pattern B. And so the way we do this in our models is the cell over here, let's say a cell over here will form connections to some subset of the cells over here. So like maybe it'll form connections to 20 of these cells. Maybe there's 1,000 active here or something like that. And when it sees that pattern of 20, it'll fire. We'll talk about how that's going to occur on one of these dendritic branches. So I might have 20 synapses here, and if they all become active at the same time, then this guy's, I'll talk about how that works in a moment, but he'll say, oh, I recognize that pattern. So, but you want a population here to be invoked by a population here. If one of these cells here could make 20 synapses on one of these cells, then this one cell will make that cell become active. That's not what we want. You just don't want that. You want a population to a population. because this particular cell is going to be active in many different combinations over here, and I don't want one cell here to drive it. You just don't want that to happen.
Wouldn't that be a useful way to code like kind of strength? Yeah.
But it doesn't do it. It doesn't happen.
And my other question was okay, in the same dendrite segment, you don't have multiple connectors to the axon, but what if you go to like a dendrite segment?
You can see that. That does occur. And so let me just jump to the sort of conclusion here. So our model, and I'm pretty confident this is basically what I'm very confident is going on. What happens in, well, I'll give you, I don't know if I'm going into too much detail on that.
No, go into details.
Okay. So historically, when they looked at neurons, the original thought was, oh, it's like this, right? I have these inputs come in and I sum them up at the cell body. And if I have enough of these inputs, then this thing's going to spike. And then when they did some studies, and they said, okay, what happens if I activate a synapse right here? I activate this one. I can just activate that one synapse. It says, oh, that's good. It raises the voltage a little bit at the cell body. And if I activate enough of them, then the voltage of the cell body was high enough, and then I get a spike. What happens if I activate this synapse? Well, locally here, it raises the voltage. But over here, almost nothing happens. So if you have an electrical engineering background, these are like wires with leaky capacitances. And so what happens is that the spike decreases as you move along here. It's leaking out to the membrane, and there's a impedance basically loss, and so it looks like if I activate this synapse, nothing is going to happen here, or so little that it's barely worth it. And then you could say, well, what if I activate 100 of these guys all around here, and you look at this hardly anything. It's like, well, these synapses can't be doing anything. What good are they? Right? Because even when I activate 100 synapses around this cell, nothing happens. And so this is kind of a mystery for the longest time. And there's a whole history about people studying this. And the solution was discovered, which is the following, and this is getting kind of messy now. Here, I'll draw another picture. The solution looks like the following. Here's our cell body. Here's our dendrites, right, coming out here. And then here are all these synapses. Typically, there's about one synapse per micron along this length here. So they're really packed in, really tight. You know, there's like right along here. And what happens is, if you take a section of this, which the number typically used is 40 microns, so it'd be 40 synapses long here. If you activate, let's say, 20 of them, 20 active synapses, each one is very stochastic, you know, releasing a little bit of this, a little bit of that. Then locally, what occurs, locally the voltage gets raised enough here to generate a spike in the dendrite. And it's a different type of spike as this one. It's not the same chemical composition as this one here. So it works on the same principle, but it's a different, it's actually, this is like spike A and this is spike B type of thing. So this is called a dendritic spike, and this is now, this is an axonal spike or a somatic spike. And so individually, if I activate any one of these synapses, it wouldn't do anything here. But if I get 20 of them so active, it generates a spike, and then the spike travels along here, and it gets to the cell body. So, and now it has a big effect at the cell body. So this becomes like a pattern recognizer. If I can recognize a pattern here, then I can create a significant event here. And that occurs anywhere along these dendrites. So there's lots of 40 micron segments that you could activate 20 so synapses, and then you'd have an effect at the cell body. But if you just activate a few here, and a few there, and a few here, nothing occurs.
So I could have one synapse at each dendrite segment connected to the same axon. Would that be okay?
It wouldn't have any effect. It might be okay theoretically. Yes. I don't think we'd actually even see that. But let's say this is a bit controversial. The question about how many synapses can an axon make on a cell. I've seen studies that say a particular axon will make one synapse on a cell. I've seen others that say, oh no, I've seen a dozen synapses on that cell. But very rarely do you see anything close to one another. Excuse me. I just want to make sure that's not important. No, it's not. Again, in neuroscience, there's all this conflicting data. But theoretically, it makes no sense. If what we understand now, it theoretically makes no sense for a single axon with excitatory synapses, not inhibitory ones, excitatory synapses making multiple connections here because it blows out the whole concept of pattern recognition. Now, the whole idea, everything in the brain goes from a sparse pattern to a sparse pattern to a sparse pattern. And invoking some representation evokes another representation. That can be in the same set of cells, like a sequence memory, like the same set of cells go from this activation to this activation, or it can be across from two populations in the brain. This is pretty much everything we talk about in most of the communications in the brain like this, between sparse patterns and sparse patterns. And this all works now by the fact that each individual neuron is recognized, each segment is recognizing a sparse pattern someplace else. So if I want B to be invoked by A, all these B cells will have one of their dendritic segments forming connections to 20 or so of these cells over here. So when these 1,000 cells become active here, all these cells will have a dendritic spike that says, oh, I recognize that pattern. But an individual cell here will participate in many different patterns. So I can't say what this particular cell means. It's going to participate in pattern B, and it can participate in pattern C and pattern D, and it can say, oh, this is going to I'm going to recognize A here. And then another time I have X and that goes to C here, and it says, oh, I'm over here, I recognize pattern X over here. And so this cell will participate in many different things. And it's the combination which really leads to the difference. You always have a different population code. Individual cells don't tell you much. It's a population code. So let's just we're almost done here, unless you want to talk more about this. Oh, I haven't talked about the one last thing. Just filling out the rest of this here. Remember I said if you had enough synapses active close to the cell body, not on the cell body, but close to the cell body, it'll generate enough here that I get an action potential and it goes out on the action, right? So enough input locally will make this thing spike. When this occurs, and I get this dendritic spike, you might think, oh, that should make this spike too, but it never does. It just doesn't happen. What it does is it raises the voltage quite a bit, but not enough to make the spike fire, the cell fire. So when they learned this, it was like, what the hell's going on here? It's like, finally, I have way of doing something with these synapses out here. And so I'm recognizing that, but it doesn't generate an action potential here. And after a little while, this goes away again. So what good was it? Right? It's like, what's the point of that? Well, our model, basically, the paper that's labeled Why Do Neurons Have Thousands of Synapses explains really for the first time what we think is going on here, is that this sub-threshold depolarization, right? It's not enough to make it spike. Has an important effect. It's like a prediction. It's saying this cell is almost ready to fire. It's on the edge now. You've given it the ready set. It's ready to go. And if it gets any input here from the side, it's going to spike a little bit sooner than a cell that didn't receive this input.
What if I don't get any local inputs?
Then it goes away.
But if I get input from several dendrite segments, can they sum up and?
I don't think that's what people originally thought. They said, okay, well, if one of these isn't good enough, maybe I have to get them from all these different ones, right? I'm not aware of the literature that explored that further, but I don't believe it's happening, okay? And I don't know I know of no evidence that that's happening. It was pure speculation because they're saying like, we got to have something happen here.
So the spike that's moving down the dendrite is an NMDA spike.
Yes. Well, there's also calcium spikes.
But so it's fundamentally different than the action that moving.
Yeah.
But the depolarization of the soma is just a depolarization. It's just a voltage.
Yeah, but
the actual state of the soma is
yes.
the same regardless
Yes.
of where it comes from, where the action. Is that right? how's that?
Yes, there could be some differences. The depolarization is a depolarization. I'm trying to remember if there's a difference in the duration of the depolarization. For example, Marcus, remind me if I remember this one here. There are these. There's possibility under certain conditions where the local depolarization here is increased longer. And there's a possibility, I remember, there's a possibility that when this spike arrives here, it stays depolarized longer than local depolarization. I don't remember.
We think that the dendrite is producing a burst or a one spike and passing that information down and not continuing to produce spikes until a confirmed, like it's producing predictions, right?
Well, our interpretation, we accepted the neuroscience literature on its face value. The neuroscience literature says these dendritic spikes, these NMDA spikes occur, they depolarize the cell almost enough to make it fire, but not enough to make it fire. These inputs do make the cell fire. And then we said, well, what? If I was depolarized because of this, and then I get this input, then the action potential that occurs, so imagine here's a little timeline. Here's what, when this input comes in here, this thing would spike like that. And what we're saying is if it's been depolarized, because it recognized the pattern first, what's going to happen is it's going to spike a little bit sooner than if it would otherwise. And that has a big effect. It may not seem like a big effect. It can only be, it only be a few milliseconds. But now one more digression. We're getting off of just purely neurons here. Remember I said 20% of the cells are inhibitory, right? So if I look at, there's a bunch of pyramidal cells here, all packed in together. And then the second most common type of cell, well, the most common type of inhibitory cell is called a basket cell. And there's inhibitory cells. And the basket cells are interspersed. Everywhere you find pyramidal cells, there's basket cells. There's fewer of them. It's not 1-to-1. It's more like a 5-to-1 ratio, something like that. And what's the characteristic of the basket cell? Well, basket cells get input from pyramidal cells, and they project to multiple pyramidal cells nearby. The basket concept, because these outputs of the cell like a little basket, they're sort of connecting everything in a little basket around. So what's going to happen here? If one of these cells spikes, and by the way, inhibitory cells classically, and especially these, are extremely fast. They're like super fast responders. So when this guy gets a spike from this pyramidal cell, just one input, it spikes immediately and inhibits everybody immediately. It's like saying, you're all silent now. But this guy won. And we actually believe that what's going on here is that he gets in training with this guy, so this guy is not inhibited, but everyone else gets inhibited. And so if you have a series of cells like this, and if one of them spikes a little sooner than the other ones, it shuts down everybody else.
So all the inhibitory synapses that we talk about in the neuro model,
Yeah.
are all of them coming from baskets?
No, there's different types of inhibitors. Remember, there's many different types of inhibitory
Okay.
cells.
So
And
some
remember
of them are coming
I said there's some that are on the beginning of the axon, there's
Yeah.
some of the cell body, and there's some along the edges here. The field of inhibitory neurons is very complex.
from, so like which one of these are coming from baskets?
I think the basket cells are the ones that connect to the cell body. They're not the ones that connect out here, I know that.
Okay.
I don't think they're the ones that connect on here. I think those are the chandelier cells. I think the basket cells are on the cell body. So basically, this is another sort of like basically telling everybody else, you're not going to fire.
Okay.
So we made this stuff up, but there's also, we didn't make it up. All these ideas that we use in our model have existed in the neuroscience literature. No one really kind of put them together. So this fast inhibition is something that everyone knows about. I think that the thing we came up with was understanding how this dendritic spike, his goal is not to create this guy to spike, but to make him spike earlier when he is going to spike. And then using the inhibition to basically create sparse representations. And then there's a whole mini column hypothesis, which we were talking about this morning about that. But so let's just review, because I didn't want to go on too long here.
Can I ask one question before we move on? Is there anything special about the refractory period of that 20?
This section here, okay, the refractory period refers to the time, once the cell spikes, it can't spike right away again. It needs to recover. It's like, oh, I'm tired. And then has to build up its ions again and, and get it ready to go again. And that delay is called refractory period, because you just can't spike again for a little bit period of time. The term refractory period usually refers to the somatic spikes. I'm trying to remember, Marcus, do you remember if we've talked about refractory periods over here? I can't recall.
My source here is mostly you and pseudotite, and it's on the order of hundreds of milliseconds.
So I would think there'd be a refractory period here.
Yeah.
You know?
So
Or rather, it's like the depolarization is for on the order of like.
Oh, yeah.
And then that's what I know.
So turning that around, that means that there is a significant period of time to acquire all of those firings or these
20 here?
Yeah.
No, the way this works is, first of all, the general rule is roughly 40 microns in distance. That they can't be further apart than that, because then they don't sum properly.
Okay.
They have to be near each other so that they don't leak apart.
Got it.
They also have to become active within a fairly short window of time.
Okay.
Shorter than you might think. It's like, I think on the order of like 5 milliseconds. So I mean, I have to have these 20 active synapses within, 20 or more. In some places show 15. So, but they have to be active within like 5 milliseconds of each other, otherwise they don't sum up properly. So how do you get them all to be active within 5 milliseconds of each other? That's really tricky. One of the things we think is going on is that in the brain, everywhere in the brain, and we don't have to model this, there's these oscillations that are occurring where these cells are firing in these oscillatory cycles, and there's different cycles. And one of the reasons for that is there's other reasons too. One of the reasons is to get when a bunch of neurons are going to fire, you'd like them to sort of emit, if they're all going to be active, you'd sort of like them to emit a spike around the same time, so that then they would sum up over here. So it's sort of, again, it's a very precise timing thing. And if they were just sort of, even if they're spiking at, let's say, 50 hertz, which is every 20 milliseconds, if they were randomly distributed, then the chance of me getting these within 5 milliseconds is pretty low. But if they're sort of all firing in synchrony, boom, 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 then they'll kind of arrive here at the same time. So we don't model any of that here. We don't think we need to. We basically just, there's a lot of biochemical stuff going on we don't want to model. So we don't model that. It's not necessary. There are other oscillations we might have to model, but not that one.
Even without the clock and the oscillation, you can see how a rate code might be useful in probabilistically producing these like synchronized action potentials, because a really fast code is going to be more likely to come in at the exact same time.
Yeah, well, close enough, but yes. I'm not disagreeing with Jeremy. That's all true. One of there's something to keep in mind. When they first started looking in the brain, but even today, so this is like 2.5 millimeters in a human, and maybe 1.8 in a mouse or rat, something like that, the thickness of the cortex, right? And they stick this probe in the cortex, and they're trying to find cells. And they're trying to find cells that are active. So this goes back to the 1960s of the work with Hummel and Wiesel. They have this cat, and they show the cat some image, and they would try to find what the cells respond to, right? So this is where they learned that cells respond to edges and things like that. Well, what they did is they kept moving this probe detects there's neurons all over the place here, thousands and thousands of them. And they kind of stick this probe in until they find some cell that was firing really actively. And they go, ha, that cell represents, a line like that. But along the way, they found lots of cells that were just like, they would spike every once in a while, just one spike. Boop. Then it was that, later, boop. And they call that background firing rate. They found even more cells that were silent, that never spiked. Well, what good is a cell that never spikes? It's like, come on, that's not right. Well, it turns out that the vast majority of these cells spike so infrequently that they just ignore them. They can't, I can't correlate it easily to this input. But then later analysis showed that even though they can't figure out what they represent, they are correlated to the input in a broader sense. We saw a nice paper talk at Coastline a couple of years ago about this. The woman talked about like, hey, you know, let's talk about those 50% of the neurons that people never talk about. And she said, well, find these spikes every once in a while. And then they did this sort of correlation and then found like, yes, with high level concepts, those cells seem to be correlated with things, but you know, it's not obvious because they're looking for something that's going. So again, there's some cells that seem to be rate encoding, but there's a hell of a lot of more cells that don't seem to be doing rate encoding. They're doing more like this very sparse activations. So I'm not saying what you said is wrong, but we have to not fool ourselves with thinking that most of this stuff is working by rate encoding. It's not. There's a huge number of things. The whole thing is really mostly built on, not completely, but largely built upon sparse populations, activating sparse populations. And it's all about the precise timing of those populations. Yeah, but that can't be true. It's not true everywhere. There are many places where the actual rate code does matter, even like grid cells. It's a very important thing, grid cells. But so it's a combination of these two. But we have to understand that. So when we thought about the sequence memory, which is very fast and very precise, when you hear someone talk, the cortex has to differentiate these patterns that are occurring on the order of 10 milliseconds. The sequence that's coming in is happening on the order of elements that have to be differentiated within 10 milliseconds of each other. And there isn't a lot of time for all this sort of rate coding going on and the next rate coding going on. It's going to be very precise pattern. It's going to be like a sequence memory, pattern, really rapidly. There's no time to settle out and average them up. You know what I'm saying? So it's complicated because there are places where sequence the temporal coding, rate coding is important, but it seems to be more places where rate coding is not important. It's more about sparse patterns, activating sparse patterns. So anyway, what we think, so what are these things we have to pay attention to in our models? It's not clear. I think the whole idea of the neuron making a prediction is so central to the cortex, meaning almost 90% of the synapses on these neurons are predictions. They're recognizing patterns for predictions. And so 90% of what the neuron does is trying to predict its input. And so it's really kind of hard to imagine how you could build a network out of these kind of neurons that really does the prediction the right way. Most of these predictions are silent. No one ever knows them. They're just like it's like if something wrong happens, you know it, but if something right happens, you don't, it just seems normal. There are other types of predictions too, but most of the predictions that are going on in the brain are silent. They don't actually leave the neurons. They're actually happening inside. And so it's sort of part of the fabric of the whole thing. It's very difficult to know what parts of these you can separate out. Can you separate out the predictive neuron from the active dendrites to the population codes, the sparsity, and so on? So I think what we're trying to do now, what Supersize trying to do now, is just like, let's just focus on the sparsity aspect first. We're going to do that. Let's go towards more of towards the binary synapses. Let's go to learning by growing new synapses, changing the connections. Those are all the levers we can turn here. It's not clear what's the right pathway to go, but you guys are figuring that out.
Another question about active dendrites.
Yeah.
It seems like, or I guess the question is, do you think it's necessary for the dendrite segment that produced the prediction to know that it was successful in
Yes. Yeah, it is. Yeah. So it gets complicated. So when this cell actually emits a spike, right? A real spike, a somatic spike. What happens is it travels backwards. So it comes down here and it goes this way, but it also goes out here and up here and back up here. Okay. Now it's tricky because when this spike comes down the axon, it goes all the way to the end everywhere. When it goes back to dendrites, it doesn't go everywhere. It kind of peters out along the way. Well, it could be, but here's something we do know. If this dendrite was recently depolarized, for example, maybe I had a dendritic spike, then what's called the back action potential, is much more likely to follow that route all the way to the end. So where we want to learn is we want to learn here. We don't want to learn anywhere else. This is the place where the heavy learning is going to occur. These are either these synapses help this guy make a prediction or they didn't help to make a prediction. All the other synapses, we just want to ignore them if we could. Right? So our learning rules say, and this is another thing which differs from the point neuron model. Our learning rule says when you have an active cell and you want to do heavy learning, you do it on this segment only or the proximal ones. You don't want to train anything else. Nothing else would care at this moment in time. And so the mechanism for doing that is a back action potential that reaches out to this section here. This guy knows it recently generated dendritic spike. Says, I did a dendritic spike, and within, let's say, 100 milliseconds later, or something like that, I now get a back action potential. It's golden. We're going to do synaptic plasticity.
But all of the synapses overactive.
Well,
Some
okay.
synapses that-
Well, in our model, everything here goes under learning. Some things will be, I forget, I don't actually where we ended up on this, but there's some degradation of it. You know, we do some things that didn't active or slightly declined. I don't remember.
You certainly wouldn't want to learn all the synapses that are on that segment because there's lots of that are coming from.
Yeah, although remember, it's in a very small area here, so we're really only talking about 40 microns. There could be 40 synapses there, and most of them are going to be, it's a gray area.
So that segment, you can only learn one pattern.
Well, okay, you're in a gray area. We don't know. It turns out, technically, because of the way sparsity works, you could intermix a whole bunch of different patterns right here. Let's say instead of having 40 synapses, I gave you 80. And now I could have 4 different things. I can intermix them. Nobody gets confused. Because of sparse things, they're not going to overlap. So you could intermix patterns and it won't get confused. Nobody gets confused. But you don't want to do that for the whole neuron. If you do that for the whole neuron, then things will get confused. So there has to be some amount of locality here, but it's like a union property. I can have multiple synapses. The synapses that are representing a pattern A, they don't have to be right next to each other. They just have to be within 40 microns of each other.
Does the neuroscience have any hypothesis about how this back tracing works? That sounds like.
Oh yeah, it's observed.
It's observed, but the mechanism.
Oh, it's the same. Yeah, it's the same mechanism that happens here. It's exactly as well. I should say.
It's the tracking, the pathfinding.
I don't know. I imagine there must be theories about it. I don't know.
Yeah, because the phenomenon I know about is that like these channels, the NMDA channels, they stay open for a little while. So if this back propagating action potential, even if it did go to all of the branches, only some of the segments will be affected by it. So that's what I know about. I didn't know.
I didn't, but it definitely is, because in neuroscience, there's contrarians to everything. You say anything, I can say neurons are synapses, there'll be someone saying that's not true. I'm not joking. It's crazy. And so there are people who don't want to believe the importance of active dendrites. There are people who don't think these dendritic spikes are important. And so they'll make the point, well, they can only work if the backtraction potential gets all the way there. If it doesn't get there, then how can you learn?
I guess I just thought a back action potential didn't really need anything supporting it. It just happened.
No, but it turns out that's not true. So these people point out that they get, because the physical, the propagation along, imagine you have a little cell membrane here. This is, this look either an axon or dendrite. And there's these little channels all along here, which are used to, these are ion channels. And so these have to let in ions and let out ions, there's pumps and so on. And so there's two things that determine whether this will keep propagating. One is the width of this thing, and the other is the distribution of these channels. And so it's very dependent on the distribution of these channels. And so for whatever reason, and I don't remember why, this is Jeremy's question, I don't remember why, but they point out that an back action potential typically will peter out and not reach the end of the dendrites. So then they say that it can't be very useful, because if it doesn't reach the end of the dendrites, then other people came back and said, oh, but we measured it, and when there was a recent depolarization, it does keep going. So they're back and forth about this, they're arguing with each other. So look, it seems crazy to me that all the neural models in the world have no accounting for the thousands and thousands of synapses that these cells have. They have no accounting of the fact that these synapses don't seem to sum up the cell body. They have no accounting of the fact that there's these dendritic spikes, which we didn't know until 20 years ago, or so, something like that. So the idea that these things aren't happening seems crazy to me. It's like, of course they're happening. This is where all the memory in the brain is. It's all in these synapses. Almost all the memories in these synapses are on the distant parts of the dendrite. These are 90% of the synapses are out here. And so they must be, that's where most of your memory in the world is occurring.
So I have a question. There's a metabolic argument that says, I think you made it in your book, where if you're recognizing something and it's the same, it's constant, you don't want to, have things firing and being really excited because I've seen this before, I can explain it. So what you're showing here is a very dynamic system that keeps recognizing things, action potentials fire, bone raws, ions are moving, metabolism is happening. So how do you balance the I don't want to waste my energy on something that is I know about?
I don't remember what I put in my book, but I don't think it was exactly that.
Okay.
There is this machine learning idea called predictive coding. And the machine learning idea of predictive coding is that you only want to transmit errors. So if you predict something, then you don't want to do anything. It's like, once you predict something, stop. That's not what I said in my book.
Okay, sorry.
And I don't think that's right. What happens here in this situation is, if a set of cells becomes active, in our temporal memory, and I'm not going to go through this today, in our temporal memory algorithm with the mini columns that Jeremy was talking about this morning, if I have predicted an input, then I will form a representation of that input that's very sparse. It says this is that input in this context, and there's very few cells active, but I'm representing it. If I get an input and I didn't predict it, and this has been observed by neuroscientists, and this is in our model, many more cells become active. It's like a superset becomes active. And it's the way we would view that is like a union of things. It says, okay, it's like I'm listening to a melody and I'm predicting the next note in the melody. If I get that next note, I will form a very sparse representation of that note in the context of that melody at this point. That's what it represents. If the wrong note comes in, then the cortex is going to represent it, but it's going to represent it almost like a whole bunch of cells are going to become active. It's sort of like saying, well, it could be any song now that has this note. Let's start over again, right? It could be any song. So let's refresh and start tracking now where we might be. So you'll have much more activity on an unpredicted input, much less activity on predicted input, but we don't believe in the predictive coding concept that you don't pass on anything. It's not like you're always passing on something. It's either you're passing on the exact thing, you know what it is, which is a very sparse, low energy consumption thing. Okay, this is cool. I only need a few neurons active to represent this. Or like, oh shit, something happened. I don't know what it is. Look at this. And then all these cells become active at once. And that's what's seen when you have unpredicted input in the cortex. When that occurs in an animal looking at a visual scene or anything, you get a much higher activity all of a sudden. And in our model, that's like a union of all possibilities. And it does other things too, but so it's not like it's silent. It's never silent. These things are always happening. It's just that if you try to look at these cell populations, it'll be very sparse, very sparsely activated. It'll be very difficult to see. They would look like they're just sending a single spike, but it's a population that's doing it, and so it'd be very easy to miss.
So I'd like to get a sense for whether we should be trying not to use negative weights or learned negative weights. And I just want to bring up the argument as like, I'll just give an alternate picture of a point neuron people probably drawn things like this before, but I'm just kind of making this up on the fly. But if you think of a point neuron as, having its inhibitory part, and then we're just calling this a point neuron. And like you have learned connections onto both of these. I'll just draw that on circles, and learned connections onto this as well. Circles. This is the same picture as that, just drawn to look a little bit more biological. And so I think a lot of us, when we think of negative weights, we're kind of thinking of some equivalent of this picture, and we're just abstracting it away, and that's what a negative weight is. How okay are you?
A couple, not very good.
Okay.
A couple of things going on here. First of all, this inhibitory neuron inhibits lots of other cells. It's not attached to this guy, right? So this mechanism I talked about, where you have the immediate spike and then like erase it here, like these guys, right? When the inhibitory cell goes, it shuts down everyone else. That's not what's going on here. This guy would say, oh, I get, this guy's recognizing some pattern, and if it sees that pattern, it shuts this down. No, it has to shut down a lot of stuff. So it's not associated with this guy. And the second thing is, most of these inhibitory cells, not all, but most of them, if I think about the connections from the pyramidal cell to an inhibitory cell, that is not, on the ones I just talked about, the basket cells, no one believes that's learned. That it's like every pyramidal cell makes a connection to an inhibitory cell. It's not a pattern. It just says, if you spike, I'm going to spike. And it's like a 1-to-1 or a many-to-1 type of thing. And so none of that's captured in here. It's this network properties that you can't get there doing it this way. So I don't believe that's the case. And so these synapses, on this particular case, the basket cell, as far as the connections I was just talking
They're
about,
definitely not basket cells.
well, they're not learned. If this is the fast basket cell, then, well, basket cells are the number one type of inhibitory cell. So maybe some other inhibitory cells might be like this. I don't know. But in general rule, inhibitory cells always make connections to lots of pyramidal cells. Whether that's bipolar cells, or whether it's chandelier cells, or whether it's basket cells, they make connections to many inhibitory cells. Generally, make connections to many pyramidal cells, and so it's not like you can assign one to this neuron. Well, like that, it doesn't look like that. So I don't think, I don't, it's a very good question. Well, how bad is it they're doing inhibitory neurons? I think the more fundamental problem in my mind is that the point neuron has no concept of prediction. And there's no concept of being able to read. It's very difficult to form these sparse representations that are meaningful, that one sparse representation invokes another sparse representation reliably. We're doing it, but it's if I take 10,000 neurons, as we've shown here, this thing can learn, and I have like 1% of the cells active at a time. You can represent an infinite number of things practically, and unlimited number. And because you can say, how many, the 10,000 is like how many times did I pick 100 out of 10,000? It's like incredibly large. And then you can learn hundreds of thousands of transitions reliably in that 10,000 of cells. It's incredible. It's really high capacity. And because these cells just don't get confused. They just, every cell says, oh, I can belong to hundreds of different patterns, and I can participate in hundreds of different patterns, and everyone does it, and no one gets confused. It's always the population that matters. Any individual cell will be, let's say, be active in every 50 or 100 patterns, but the combination is going to be unique to trillions. So it all works out nicely, the mathematics about it. Nothing gets confused. Anyway, so I think to me, one of the largest problems here is this idea that to build these kind of associative networks that work really reliably from point to point, or high frequency, high dimensionality, I don't know how you separate out having a predictive neuron and not having a predictive state in the neuron. There's no prediction here. Either the cell is active or it's not active. There's no other separate thing going on. You might have to have a separate population to be a predictive cells or something like that. So that's one of my big bugaboos about the point neuron. And then I think the whole idea of learning through rewiring is so much more powerful than learning through synaptic weight adjustment. So, and all these things play together in some sense. So it's difficult to know which parts you can leave out at the moment and try to make it work. I think you're doing a good job, you as the team, about exploring the sparsity aspect as its own thing. But ultimately, I think you're going to need to get to this sort of idea of a multi-stage predictive neuron.
Can I ask a question on the dendritic segment? So the dendritic segment spikes. So it generates some voltage, right?
Mhm.
And let's say that neuron doesn't spike at t equals 0.
Well.
And then at t equals 1, if there is no inhibition, that voltage is still going to be there. It's going to be less, but it's still going to be there.
It goes away pretty quickly.
It goes away very quickly. But I mean, if t is infinitely small, let's say, you could say that
It's exponentially,
Yeah, it's
More
like-
resonance.
it's decaying, but still there, right? And right now in our temporal memory model, we're only considering t minus 1, right? We're not considering fluctuations from now.
Yeah, you mean like make sure the state depolarized for 2 steps or 3 steps, something like that?
Yeah, like there should be some explanation.
Yeah, well, first of all, it's not really exponential. I'm trying to remember now. I'm trying to remember what the literature says about this. I mentioned it, and Marcus mentioned it too. There are situations where that time frame can be increased significantly to hundreds of milliseconds, and other times it's very short. So it's sort of a function of the cell and what's going on. And this is what Murray Sherman, he talks about these metapotropic receptors, that under certain conditions, this depolarization can last a long time being hundreds of milliseconds. That's not, maybe a second or two. But I think when we thought about the sequence memory, we said, okay, well, that would be sort of a way of keeping track of where you were without losing context. Like you know, in our current sequence memory, if you have one wrong input, the whole thing gets lost, right? It gets lost. And so maybe I should keep some of these synapses active for longer periods of time. And I don't know if we explored that. I can't remember.
Not while I was here.
Now I use here. There's an alternate way of doing that too. The alternate way of doing that is you have another set of cells which are stable throughout the sequence. They represent the title of the song, the name of the song. They're the classifier, if you will. I'm classifying this thing, and that classification is the same throughout the entire sequence. And so that classifier, and that's called our temporal pooler, and that classifier can say, hey, I'm listening to this melody. You know, it's a national anthem of Brazil, whatever that is. And a wrong note comes in. I don't forget that I was listening to the national anthem right away, right? Maybe in 3 or 4 wrong notes I'll forget, but right away, now it's still there a little bit. And I said, nice, I was listening to that song, and then I'll hear it starts up again. So my guess is that's more likely to happen in the sort of temporal pooling layer, where it's saying, you know, I know what song I'm listening to. It's just, I have a stable representation for that, and I'm feeding that back onto this network, and we have modeled this. And so it says, yeah, you can get out of step for a little bit, but I'm still know that I'm supposed to be listening to this song for a little while, and I'm not going to forget that right away. But if I have enough wrong notes, then you kind of forget it. And it's like, yeah, I'm not listening to that anymore. I'm now listening to something else. We don't really know the answer to your question, but I think it's more likely to be in the temporal pooler layer than in the than sort of an extended duration of the predicted state.
But it wouldn't be biologically plausible.
No.
If we.
No, it wouldn't be like, I mean, I think it's more theoretically troublesome. I have to think about like, what's going to happen, you know? So now you're predicting. Imagine I kept my predictions active for 3 notes. That means if the next note in the sequence didn't occur, but the previous note occurred again, then I wouldn't notice it. How'd that be? You know, like. But then I could just keep repeating the same note over and over again. It was like, you know, it would say fine. I don't know what it would be. My point is, if I'm making multiple predictions at the same time, then any one of those predictions would occur. If any one of those inputs occur, the system is going to think it's good. So it wouldn't know if it was wrong. I come to sort of believe that this prediction stuff is very, very precise, meaning it's very temporally precise. And it gives it the ambiguity by forming these unions, as you probably know. But it's possible that you know, but you'd have to deal with the consequences. If I kept it going, then I wouldn't be able to tell if the next note was the same as the previous note. You know,
If you're using binary, if you're not using binary, then it could be some sort of decaying. You know, you still have that signal, but that signal is a lot weaker.
Yeah.
It's just an exponential decay of memory.
Oh, okay.
So it goes down by 0.5%, half the second time.
yeah,
It does result in these problems of repetitions because the old predictions are still active, and so it continues to predict.
yeah.
And yeah, I'm not sure how to do with occlusion, right? That's the problem. Like you miss the note.
Yeah, well, I think the temporal pooler does that. Again, we don't have this all figured out, so I don't want to imply that we have all this stuff figured out. We have working solutions of all of it, but some of it's not quite right. So the way we've modeled this is you might have this, this is your temporal memory layer, and this is going to learn sequences. And so there's always a sparse pattern here, and it's really good at the memory sequences. Then you can have a temporal pooling layer, which is basically stable, sparse, and unique for the sequence. So as this thing's changing, bing, 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 this guy is just sort of, is like the name of the song. And there's a limit to this because I have to map multiple patterns here onto one pattern here, and there's a limit to how many you can do that. But under this scenario, this guy can continue on saying, yeah, I know what melody this is. And he could be driving back here and saying, you know, you're still listening to that same song. And so think about these, like these apical dendrites here. He says, you're still listening to the same song. Actually, roughly, it'd be like this. I have a pyramidal cell here. It has its apical dendrites up here. And so this guy is saying, okay, I'm still telling you, you're listening to this song. And these synapses here are saying, what's the next note? So the next note comes wrong, but this guy says, yeah, okay, that was wrong, but you're still expecting this melody here. And so there's still a bias in these cells for what the next note should be. So that's how I think it's more likely to be solved in the brain. And we have models of this, and it works. This is actually a bottleneck because you can't. An interesting thing here, you can't. There could be millions of elements here, hundreds of thousands of elements here, but you can't map a sequence with. You can't map all of those onto some pattern here. It's just too many. These cells have to recognize many different patterns down here and say it's part of the same song. And if you think about like trying to name the tune, if you listen to a melody, it's easier to name the tune if you're in the refrain, the part that repeats over and over again, or it's the title of the song, something like that. But if you're in somewhere else in the song, you might, I know that melody, I know it, I can't think of the name of it. It's because this guy knows it, but this guy hasn't been able, this guy can't remember everything. So this guy's going to learn the most common subsequences here, and that's the refrain. So when you get back to the refrain, this guy says, oh yeah, I know that. But if you're in the middle of the song here, it's this guy says, I didn't learn that. You know, that's too much for me to learn. So, maybe that's what's happening there. So I think that was I'm happy to answer questions, but I have no more material for this. I didn't want this to go on this. I didn't want to keep you all busy for this one.
That was awesome.
So if you read the why does the brain and why do neurons have so many synapses, talks about all of this. And Subotai did a really nice analysis in that paper about the mathematics about these synapses, why it looks like the brain requires about 20 of them. And Subotai showed that that's sort of optimal mathematically under a broad range of assumptions. So it was a really nice theoretical derivation of a biologically observed phenomenon. I'd encourage you to read that paper, and that covers everything I just talked about here today. I think it doesn't cover the inhibitory neurons too much, but it covers this plus a bunch of other stuff. That's an important paper. I think you should all read that if you haven't yet. Okay. Maybe next time we'll do the temporal memory.
Thank you.
Yeah, sure.
All right, thanks, Jeff. I'm going to stop the stream.
Okay.
Very well.
Thanks, everybody.